Let's begin with today's a new lecture. Hello guys, a very warm welcome from the Spice of Your Biology. Today I have taken a very unique topic that is called as termination of transcription. Here will deal about both the prokaryotic as well as the eukaryotic system. How exactly the transcription process gets terminated in the prokaryote as well as in the eukaryote. Let's deal with it. I hope like you're going to watch till very end, and this is going to give you a lot of information regarding this process. So the name of the topic is termination of transcription in prokaryotes as well as in the eukaryotes. Let's first talk about the prokaryote, where exactly the prokaryotic transcription terminates. So in the E. coli, we have the two common features where exactly the RNA polymerase comes off from the DNA template. First, there will be 4 to 10 conjugative AT-rich region, and A is always going to be on the template strand, and transcribed RNA is terminated or in just past the sequence. Another one is the GC-rich region, which is a palindrome. Palindrome is a one sequence which can be read, from both sides. They have the same words, and that is going to be immediately followed by the AT-rich region. Let's try to see that. Now you can see over here when this sequence is going to come. So when the RNA polymerase moves, it makes a hairpin loop, and to terminate this process, we require the strong hairpin loop formation. There'll be two ways: one the row independent, another is going to be row dependent. This is one which is a row independent. When the strong hairpin loop formation occurs, in that case the termination of the prokaryotic transcription is going to take place. This is the sequence I was just talking about. You can see the GC rich region and the followed by the AT rich region. Okay, and you see this is the hairpin loop formation which is going to happen. If you just look at it, this part, a lot of GC is going to be involved in making this, and this is going to make the strong hairpin loop. If you look at the percentage, and is going to be very high, and if you delete these portions like the C 41%, A 25%, U 20%, and G 14%, there's a one side because the right side has a sequence which is rich in the C and poor in G, preceding the actual site. And if this is going to be terminated, you can see over here, so the termination is not going to take place. Look at this region I was talking about, GC rich region, that is going to form the strong hairpin loop. This is called as an intrinsic termination, and this is going to vary. This hairpin size is going to vary from 7 to 20 base pair, and the stem loop structure includes the GC rich region. Just now I spoke about. Now let's talk about the row. Row is a helicase which is going to bind to a site we call the rut site, and you can see over here this row is going to translocate along the RNA. It will move around, and the RNA polymerase pauses at a hairpin loop, and row is going to catch up. Look at over here, there's a lag between the movement of the RNA polymerase and the row, and as the hairpin loop comes, it slows down, then this row is going to catch up, and row is going to unbind the DNA and RNA hybrid, and this will lead to the termination of the process. So termination often requires the row factor, just now I told you. And what exactly is the row factor? Row factor is a helicase which unbinds the DNA and RNA hybrid. Even it can unbind the RNA double helices as well. This requires, like all the helicases, requires the power from the ATP. And genetic manipulation, if it's going to happen in the row dependent, the termination requires a specific recognition sequence on the nascent, which is just upstream from the termination site. If you go into the structure of this row, which has the two domain, you can see the C terminal domain and the N terminal domain. N terminal region is going to bind with the RNA, and C terminus is going to bind with the ATPase, which has the inbuilt activity and that generates the ATP hydrolysis. And this is how this process is going to take place. Now let's compare one wild type, another one is a nonsense mutation. Ribosome, look at over in the wild type. Ribosome pack messenger RNA behind the RNA polymerase. You can see over here, this is the ribosome, and the ribosome impedes row attachment as long as it is bound. Look at over here. So this, the row will be always behind the ribosomes. Okay, so it will not get a chance to move further and transcription continues. Whereas if the nonsense mutation occurs, that means a premature termination of the protein synthesis. Let me remind you that transcription and translation in the case of prokaryote, it goes hand in hand. So here if we just look at, if the ribosome detaches, so quickly the row is going to catch up, and that leads to the termination of the transcription process in the prokaryote. Let's go to the Eukrotic process, and we'll start again from the CTD domain, C-terminal domain of the RNA polymerase, which is very, very important because all the processing enzyme, which includes the capping factors, splicing factors, and polyadenylation factors, which are there. And as soon as, if you look at, they will get a chance once the nascent RNA is being formed. So capping factor, splicing factor, and the polyadenylation, they're going to bind. And as the process stops, terminates, they start doing their jobs. Let's not go into that. Splicing sometime else we'll discuss. Let's see how exactly the 2-3 points I would like to talk about. One, how exactly it gets a signal to stop the proceedings. Second, how the transport of the messenger RNA occur across the nucleus to the cytoplasm, and how the turnover number of the messenger RNA, how long a messenger RNA is going to be leaving. That is, the another part that I would like to talk about. Let's go one by one. Look at this. Once the cap is formed on the 5 terminus, and you can see over here. So this is the one protein we call the CBC, cap binding complexes, which is going to protect this on the 5' so that exonucleases cannot attack it. And this plays a very important role even in the transport of the messenger RNA from the nucleus to the cytoplasm. Now that the few factor which is going to play a very, very important role. Look at this. One is the Cpsf, that stands for the cleavage and the polyadenylation specificity factor, which is, you can see over here, which is going to bind to the this particular location, that is called the AAUAAA. Then we have the CSTF, that is a cleavage stimulation factor, which you can see bind over here. And another enzyme, we have the polymerases, because a PAP, and this is one which is going to do the job of the poly A, the polymerase, which is going to put the tail end, the 3' end, which is going to put multiple adenine residues. And immediately after that, the poly A binding protein is going to bind to those locations, which is very, very important. And when this is going to happen, RNA polymerase eventually terminates. It just look at what this region where the poly A signal encoded into the DNA. Though there's no specific boundary between the two genes in the eukaryotic system, but there are certain signals which are there, which plays a very important role in the termination of the transcription process. One is this one. Now, as I told you, polymerase comes, and certain additional factors. Polymerase require no template strand required for this. Okay, and followed by the poly A binding protein. I told you the site which it requires. And once the poly A length is going to be there, this length is going to be very, very important. All of A nucleotide, all they derived from the ATP only. And you can see this 3' end where the A is bound. Look at this 200, the A residues were shown over here. That could vary. And here the poly A protein binds, and which prevents the messenger RNA to undergo the degradation process. Similarly, polyadenylation is going to be held by the few proteins, Cpsf, CSTF, CF, PAP, and the PAB, poly A binding proteins. Just now I told you, they're going to play a very, very important role in the transport, in maintaining the stability, and the translation to take place later on. There's one little information. Cleavage of the transcript past its AAUAAA sequence does not in itself terminates the transcription, but certain cases, especially you see in the yeast, there's a protein we call the RTT103, which binds to the phosphorylated CTD domain of the RNA polymerase 2. What is it? Actually, C-terminal domain, which needs to be phosphorylated in order to convert initiation factors or the initiation process into the elongation one. It recognizes this AUAA and they recruit a specific exonuclease, which is called as a RAT1, and the same one in the human being we call it the XRN2 in human. And they follow a torpedo model, which is highly processive XRN2 loads on the newly liberated uncapped 5' end of the still nascent RNA and rapidly degrades it until it intercepts the RNA proteins or the ribonucleoproteins and induces it to terminate the RNA synthesis. It has been hypothesized that it occurs in much the same way as the Rho factor, but still we need the RNAP to initiate the new round of transcription. Look at this. There's a basic idea. This is the chromatin and the certain region which are chosen to be transcribed. You can see the length of the RNA is moving. This RNA is now completely processed. That means it has gone under the capping, splicing, and the polyethylene. Now it is able to move out from the nucleus. I tell you exactly how does it happen through the nuclear pore complexes. So where once it goes into the cytoplasm, and that will be followed by the translation process. But only the few selected RNAs that can be transported, which is complete in all regards. See this, there's a huge number of the proteins which are binding to this. You can see the HNRNPs, poly A binding protein, CBC, SR, which plays important role in the splicing as well. And you can see the nuclear pore complexes, which has the FG repeats. The two amino acid, one is a phenylalanine, another one is a glycine, which is scanned through. And you can see the many proteins that are going to be, dropped down into the nucleus itself. And one of the few proteins which are going, including the CBC, you can see the poly A binding protein. There are the few more proteins which is helping it out in the process. And as soon as it moves out, immediately the initiation factors, which is many in the case of eukaryote, and you can see there's a loop formation, which is a must for the, you can see over here the tail and the protein bound to the tail and protein bound to the cap. They interact to make this loop, and then only the translation will happen. Otherwise, it will not happen. Let's see how it is going to happen. This is going to be supported by the the RAN GDP. You can see the nuclear export factor, which is the over here. It will bind to this one, okay, and then this cargo is going to be delivered, and outside if it goes, it binds with the GTP, and in the process REF, RAN GTP elongation factor or guanine nuclear exchange nucleotide factor, which is going to exchange between the GDP and the GTP. There are the few proteins that already have discussed, which is going to play a very important role in the 3' cleavage and the polyadenylation. And these are the proteins I told you, like they are going to play a very important role. Few REF just now I spoke about. This is going to bind with the exon, the complexes where we have the exon and other the proteins which is going to make this complex. And you can see the REF shown over here. And then the 2 protein tab, or we call the MEX, which is going to bind. And you can see the fibrillar region of the nuclear pore complex, which is having the lot of FG repeats, which is going to scan through it. So the few, I told you, like multiple steps of the messenger RNA biosynthesis are tightly coupled. Why? We'll discuss little later on. And these 2 proteins, the MEX67 or TAP, it is a major messenger RNA export factor, which will bind to the messenger RNA. It is a must to make it go, out of the, nucleus. And then the few more questions we need to discuss later on, how exactly the messenger RNA is going to be released and where, and the different pathways, and how it is going to be, coupled with the transport mechanically, and whether this process is going to be regulated or not. Let's talk about it. First, let's talk about the turnover number. The number of the messenger RNA required depends upon its demand. So there's a clear-cut relationship between the demand and the supply. This is what we call the messenger or turnover. The two factors, the number of the messenger RNA and how long it is going to be surviving. For example, you see this example. Here we have the high iron. So what happens is that, this protein is not, required. And if in the case, if it is, having the less iron into the medium, so this, the active factor will bind to it. It gets activated and bind to this called IRBP. Look at over here, the second, the example. This is the, messenger RNA for the transferrin, which is a protein which transport the iron from spleen to the liver. Now, the second probably will be able to, correlate very easily. That is the casein messenger. And this is the protein found in the milk. Look at over here, the prolactin. This is one hormone which works by the JAK-STAT pathway. If it is present, then the number of the messenger per cell is going to be around 30,000. Whereas if it is not there, so the number of the messenger RNA is going to be just 300. You can just look at 100 times less. Okay, but what happens is that there will be no change in the transcription. That means the excess of the messenger RNA which is formed is going to be degraded. Now there will be two ways the degradation occur. One is going to be the messenger RNA decay, which is going to be controlling the turnover number, and another is the NMD, which looks forward to that the messenger RNA should be complete in every regard so that it can be easily translated. This is for the surveillance purpose. Now, if we look at the messenger RNA decay, there will be one enzyme because of body age shortening, which is done by the deadenylase complexes, which can cut off the A. And then we have the decapitation, which is done by an enzyme because of decapping enzyme or DCP1. And later on, if the cap and tail has been removed or shortened up, you can see over here is the 5' to 3' exonuclease, XRN1 we spoke about, and then the degradation of the messenger RNA, takes place. Now the nonsense mediated, decay, if stop in the penultimate exon or the 5' splicing mark, you can over here. So DCP1 will come, and you can see over here that is going to activate the decapping enzyme and followed by the XRN complexes. So what you see over here is that this is going to work in the 5' to 3' exonuclease. Now the last part where exactly the messenger RNA is going to land up in the cytoplasm is by the certain proteins. We call it as they provide the zip code or the pin code. So, and this process is going to be facilitated by the two important, the proteins, actin and the tubulin, which will help in the process. Okay, we know about these are the cytoskeletons. For example, we have the VG1 messenger RNA, and it has to go to the vegetal pole. Similarly, the nanos or messenger RNA that's going to be on the posterior side, and the backward is going to be anterior side. So this requires the messenger RNA binding protein we call it stophin. And similarly, the post-prospero into the ganglion mother cell. This requires the stophin and mirinda. And east, we require the ASH1 messenger RNA to the daughter cell. So decay, it is going to be regulated, and the non-regulated turnover, but apparently coordinated because we have the known enzymes which is going to do that. And this can be again regulated by the some non-coding RNAs or the miRNA or the siRNAs. And NMD, it will get activated only when there will be some premature termination or the nonsense mutation occurs. And the second part of that, I just spoke about how exactly it is going to be, localized. So they have a zip code, which is always on the 3' untranslated region of the messenger RNA. This is going to be facilitated by the actin and the tubulin, protein, which is very, very important. And once the messenger RNA land up into the cytoplasm, it will be ready for the translation. And the time it is going to take, it will vary as per the demand. Not all the messenger RNA is going to have the same life period or the half-life. It varies from one messenger RNA to the other. So thank you for listening. Hopefully it has solved many purpose and probably you have taken the note of it. Watch, listen and subscribe to my channel, and many more will come in the future. Thanks a lot for listening me. Thank you.